I want to do a few things. I want to explain the standard curve, show how to enter data in the spreadsheet, and what does it all mean, and how you should analyze your data and what needs to be included in the graph. So the standard curve is based on the Beer-Lambert law. And essentially what it says is that absorbance is proportional to the length that the light travels as well as the concentration. Since we are using cuvettes that are the same size, the length of travel does not really change for us. However, what does change is the concentration. So this equation tells us that a given absorbance, if it goes up, so will the concentration. So below you'll see a graph. This graph shows the absorbance plot against concentration. So this is actually our standard curve that we got from our values. On the bottom I plotted the concentration, and on the y-axis it's the absorbance. As you see, the points make a line, which we can then use Excel to get the formula from. So before we go on to Excel, I just want to talk about what do these numbers mean. So the first thing I want to point out is the R-squared value. The R-squared value refers to how well the line, which is this black one, is fit on this scatter plot. So these points don't originally have a line, they're just points, and then Excel plots this line. And this value tells you how well it fits. So essentially, the closer to 1, the better the fit is. So so basically a point, so -1 would show a negative correlation, and a +1 would show a positive correlation. And the closer you get to 0, the more the less correlation you have. Next, I want to talk about the Bradford assay. Since proteins don't natively absorb at certain concentrations reliably, we will have to use a commercial Bradford assay. So what this does is a colorimetric assay, similar to when we use the fast blue reagent for the esterase activity. This is a colorimetric protein assay, and it's based on the absorbance shift of Kumasi brilliant blue, which is a dye. As this dye binds to proteins, we observe a shift from the red to the blue spectrum. So you will actually see the solution turn more blue from the previous brown reddish color. We can then observe the change using a photospectrometer and record the absorbance values. And then going back to the standard curve, we can have our unknowns, and we can calculate what the concentration of the unknowns are from the concentration. So if you, let's say you have an absorbance value of 0.75 for the unknown, you can use two methods. You can either look at the graph and just estimate that it should fall somewhere between 1 and 0.5, and then it should be here, and then go across until you hit the line. Once you hit the line, you go down, and the number should be roughly 250. And that is the quick and dirty way to do it, but it's not really specific. To be more specific, we have to use this equation right here. So for that, I want to move on to Excel and show you how to set up the equation and talk a little bit more about the standard curve. So this should be what you first see when you open the Excel file I sent you. Here you see a few things. You see table 2, 3, 4, and 5, and these are from your lab book. Table 5 is already filled in for you and has the absorbance values calculated during class and the BSA concentrations. So BSA concentrations were given. We got this from our dilutions because we knew that the 1x solution was 2,000 micrograms per milliliter. And then as we make dilutions, we just divided by the dilution factor. So 2,000 divided by 40 is 50 micrograms per microliter. So from this, what we can do is plot the graph. I want you to select everything, including the titles, and go to charts. And we want to have a scatter plot. So once we have a scatter plot, the x-axis and the y-axis are not filled in. I'm just going to delete this. So we want to fill those things in. For the lab report, you will need to include a standard curve graph, and it needs to be properly labeled. So on the bottom we have, okay, just give it a second while it loads. So on the bottom we have the concentration. And it's in units of micrograms per milliliter. This should not be all caps, but. And then we need the y-axis. So we'll have it here. And this will be the absorbance. And that's in AU, units of absorbance. Okay. Now I want you to go to Format, the chart layout, and add a trend line. So we want to have a logarithmic trend line because that's really the best one that will fit. So it's not here in the click, so we want to click trend line options, and you'll see you have linear, logarithmic, polynomial, power, exponential, and moving average. I want you guys to use the logarithmic. As you see, this line fits fairly well. Then we want to go to options, and we want to ask Excel to show us the equation of that line and also display the R-squared value. So that will be right here. So as we saw before, the R-squared value is 0.97, which is a fairly good fit, and I'm happy with it. So that's going to get us pretty reliable numbers. It's not as good as getting a 1 or 0.99, but it's close enough. Next, I just want to show you, let me just correct this, 971. So I want to show you how to convert the log scale, the natural log to a exponential equation. And I'll explain to you why you would want to do that. So as you see, this is a natural log equation. And what you might notice that concentration is on the x-axis. So we will be solving for x. So that means we have to move this side, the y, on the left side, on the right side, and the x onto the left side. To do this, we have to turn the natural log equation into an exponential equation. So this is essentially how you do it. I color-coded the numbers. So when you move it back, you want to raise it to e. It's y plus this number right here, the green, so 0.971, and divided by the number that the natural log was multiplied by, which is 0.3748. So this equation is already in these cells right here, the protein concentration. So essentially what table 4 shows you is that if you put in the absorbance value of an unknown, so let's hypothetically say 0.75, we talked about that one earlier. So let's say 0.75. It will spit out the protein concentration automatically. So if we go to 0.75, so we estimated it would be roughly 250. But as we see, we're kind of off by doing it this way. So 0.75 would be here, hit the line. Well, actually, this makes sense. 177 would be here. So this saying is that at absorbance of 0.75, the protein concentration is 177 micrograms per milliliter. So this is a great way for us to get the unknown concentrations of our samples. The next thing we have to do is find the activity per microgram of our different esterases. So I'm just going to put in a few random absorbances here, so 0.65, 0.77, and 0.9. So these would be the ones you collected from your samples, so your beetles 1 through 8. I'm just putting hypothetical numbers. So as you see, this also automatically filled in. but I want you to understand what's in these formulas. So during lab 1 and 2 of the 3 lab sequence, we loaded 50 microliters of our sample. You can find this protocol on page, 1 second. This was on page 70 of your lab manual. So we use the micropipetter to transfer 50 microliters of the crude extract from tube 1C to either tube 1D or 1E. So because we don't know the concentrations before we were loading just volume. So we might have been loading different amounts of protein into each tube based on the concentration of the protein. So now we want to standardize the amount of protein we loaded. So for this purpose, we will multiply the concentration of our protein sample. So beta 1, for example, is 0.75. We want to multiply the concentration of 177 by 50 microliters. So since this concentration is per milliliter, 1 milliliter is equal to 1,000 microliters. So if we want to get 50 microliters, it would be, so 0.05 milliliters is equal to 50 microliters. So just make sure you know your conversions. So we want to multiply the concentration by 0.05 milliliters. That would give us the amount of protein in 50 microliters of our sample. Then you want to put the absorbances for the D tubes. So this would be from lab 1 and lab 2 for table 3. So this is the data that should be that you should have collected during the first 2 weeks. So again, since I don't have your data, I'm going to put in random numbers just to move on with this presentation. So I'm going to say 0.67, 0.77, 0.66, 0.66, just numbers. And as you see, automatically the activity of alpha naphthyl acetate esterase is calculated for you. Okay? So then you want to fill in the absorbances for the other ones and get the data. Okay? Once you get that data, I want you to copy the all of the data back into your lab book so you have it for yourself, and then move on to the class data. In the class data, I want you to input the activity per microgram of protein into this column. So for a Zuki bean and the mung bean, okay? So please fill it into the group number that you are in. So for the 2PS section, we actually use Azuki beans, and then this was black eyed peas. And for my Wednesday class, it's mung beans. Okay? So once you fill this in, this data will automatically be filled in for you. So how do you use it? Essentially, let's think back to our hypothesis. We were looking whether the food source affects the activity of the detoxification esterases. We're using 2 separate detoxification esterases for our hypotheses, which means we need 2 separate t-tests and 2 separate graphs showing the mean. So let's just write this out. So since you have all this data here, I'm going to copy it on a separate place just to keep it neater. So we're comparing Azuki and Monk in this case. So let's put in the numbers. So the class mean was, let's see, Azuki, and here we'll write Monk. Right, so this will be for alpha naphthyl acetate esterase activity. And Azuki Mung, so let's look at the means. So the mean for Azuki would be here. So it's 0.007. And then for the Mung would be here, 0.028, 0.028. So these are the means. and then we want the standard deviation as well. So let's just write that. So the standard deviation is here, so it's 0.004, and for the month, it's 0.006. Now we want to plot this. So you want to select all of that, and go to charts, and we want let's see, a column graph, which is right here. So we want a regular column graph. Okay, so you know what? I apologize. So do not select the standard deviation yet, just the means. So you should get a graph like this. So this will show you. This will show you the means in bar graph form. So there's a few things that are missing. We are missing the error bars and the legends. The titles for the axes. So let's add it. So the horizontal. Okay. So on the y-axis, we have the alpha naphthyl acetate esterase activity for activity, and that's in units of absorbance per microgram of protein. Next, on the x-axis, we have the different treatments, either azuki or mung beans. So these are the food source. Okay. Next, you wanna add a title as well. So chart title. So we can name this NAE activity based on food source, for example. Next, we want to plot the standard deviation, which would be our error bars. So make sure to include that in your legend. So you click on error bars, and we want to do both. So that's plus and minus, and click on custom. So you click specify value, and what you need to do is actually just select the standard deviation in the same order that you selected the means. You do this for the top and the bottom, and click OK. So if you click OK, you should see that the standard deviations are plotted as error bars. So at this point, the graph is complete. With you just have to put in the legends, and besides that, everything is good. Next, so you have to do this for both alpha and beta. So make sure to make two graphs. So please don't forget that you need two graphs for alpha naphthyl acetate esterase activity and for beta. Next, you need to do a t-test. So once all this data is filled in, you will have the necessary data you need, so such as the number of samples, the number of numbers for the means, and the standard deviation. So you can use those to do the t-test. So you need two t-tests, and you need two graphs, and you need one graph showing the standard curve. So now I'm going to go over the lab report. So if you turn to page 78, you will see the guidelines for the lab report, and please follow these to a T. Also, on the preceding pages, on page 76 and page 77, you have graphs, which, so you have table 6, which is table 6 and 7, which are these two. You have table 8, which shows the T critical, and you need two of those. You need table 8, table 9. So let's just look at the lab report guidelines. You need your name and the name of other students in the group. You need a title that has to be informative. So please make sure it says exactly what you're measuring in the title. Don't say the effect of food source on um, something. Be specific. Say the effect of food source on the uh, activity of alpha naphthyl esterase and beta naphthyl esterase. Okay. Next, you have to give justification for why your experiment is worth doing. So your hypothesis should follow a clear justification that is based on research from primary literature. So make sure you look online at on PubMed or Google Scholar and find actual peer-reviewed or scholarly articles. Next, you need to include a experimental design. The experimental design should include all of your variables, treatment levels, replication, sample sizes, and species, strains, the legume seeds being used, and you may include a table to organize your thoughts, but you must, absolutely must, describe it in paragraph form, because if you don't, you will have points subtracted. So you'll lose basically half of the credit for a part of the experimental design. So I think you'll lose 6 points just for not including it in the paragraph form. Next, you need to include specific methods used. So don't repeat everything that you did, like I pipetted 200 microliters. No, but give a general summary of what was done. So if somebody that has same science knowledge as you can repeat the experiment and get the same results as you did. Next, you must say how you're going to analyze the data. So don't include the analysis there, but say what you're going to do. So saying you're going to use a t-test to analyze the data is a good way to get those points. Next, you want to talk about your results. So results and data analysis is just that. You will just show your results and you will not discuss what they mean. You will not talk about your hypothesis. This is just to present your results. The reason we have that is that, let's say you publish a paper and somebody wants to use your paper as justification for their own research. So before that person uses your paper, they will read it over and they will try to just look at your results and come up with the same conclusion you do, but independently. So it's a good way to test somebody else's paper. So if both you, the reader, who does not know what the discussion portion of the paper is, but they look at the results and they come up with the same conclusions, that's, I think, a solid paper. So you should aim for just showing unbiased results in your results section. So for that, to get full credit, you need to include the standard curve and equations for the protein assay. Don't include the raw data, but just show the graph and the equation, and you can also include the R-squared value. Next, the graphs. You need two bar graphs, with the means and standard deviations for alpha naphthyl acetate and beta naphthyl acetate esterase. So these need to be corrected for the protein content, so please don't include any raw data. The only tables you should include for this are table 6 and 7, and then the two graphs. Also, make sure you include the means and the standard deviations either in text or in a table. It's easier if it's in the table. Next, you need tables containing the results for the statistical analyses. So this is your t-test. Make sure you have two tables that look like table 9. Okay, so that's the degrees of freedom, T critical, T calculated, and confidence level. This is important for you to have. Next, one sample calculation to show how you use your data to calculate protein content and enzyme activity corrected for protein content. This is for you to explain that you know how to calculate the protein content and enzyme activity. So this is basically what I talked about in the video. You just need to show the math and explain it on the lab report. Okay? So you just need one sample calculation. So just pick one of your samples and follow it all the way through the end. So all the graphs and tables must be numbered and must have a short caption or title. A couple of sentences stating what is presented in what figure or table, summarizing the data. So this basically says you need to mention it and not just give a graph or a table without talking about it. So just say what is in that table. Again, don't discuss any of the data or like in terms of conclusions and the results. Next, we're moving on to the discussion and conclusion. So this will be 15 points, and please remind your reader what the hypothesis was. So you need to restate your hypothesis and restate some kind of justification of why this matters. State whether it's supported by the data you presented in the results section. So this is where you're going to talk about whether your t-test supports or does not support your data. Your hypothesis, I'm sorry. So your conclusions based on your data analyses presented in one or two short paragraphs. So 2 paragraphs should be enough to talk about if your conclusions are supported by the data, talk about the justification, things like that. And this is part of the justification. What do the results tell you about the physiology of the organism? So talk a little bit about the actual individual beetles and their survival and how these detoxification X-rays, how the activity might increase their survival, things like that. So just read through the laboratory guidelines. Future directions. Please include something that makes sense, something that is that makes logical sense to do next. Don't say, well, for whatever reason, without explanation, we'll get a whole bunch of new beetles, or that we'll try a different drug. That has to make sense. So make sure there's some kind of thorough thought put into it. And don't say we're going to increase the sample size or we're going to pipette more accurately. That's not a new experiment, okay? Next, you have to have the correct citation. There's just no way about it. Plagiarism will not be tolerated. So some of you already have been told this. So if you are going to use a source, you must cite it. You must put a citation in text. You must have a correct citation. So go to page, so it's Appendix A. Let's see. So page 1 of additional readings. Okay? So read that through. Make sure you're citing correctly. Make sure if you are using a source that you're not plagiarizing. If you copy it verbatim, I will know. If you're copying another student, I will know. Safety Science shows me all that, so please don't do it because it makes for a very uncomfortable situation. So this includes figures. Please don't just throw pictures into your lab report without having a citation. That is plagiarism. You did not take that picture. You do not have express consent from that from the photographer of that picture to just put pictures into your lab report. So you need to make sure you cite it and give credit to the author. Next, you have 5 points for overall organization and grammar. This is how well you put your thoughts together. If it's a jumbled mess and I can't understand what you're saying, you will lose points in this part. Most people do fairly well. Just make sure it's not a rush job and the lab report makes sense. Your writing has to be concise and precise. Any word, phrase, sentence, or even paragraph that does not add to our understanding of your project should be deleted from your lab report. Some of you had that, or actually there were some lab reports that you just copy and pasted the same paragraph twice, which I don't know if you were trying to increase the word count, but please don't do that. After your instructor returns your grade lab report, you will have the option to rewrite. So just like lab report 1, you can rewrite this lab report. And I urge you to do so because it will improve your grade drastically if you make the necessary changes, okay? Other than that, I do not have any more wisdom for you. I will post a link to a Khan Academy video that will show you how to work with the standard curve a little bit more. So it's not going to be with a protein assay. It will be with a solution of a certain compound, but the idea is the same. Once you make a standard curve of a known concentration and known absorbances, you can use that standard curve uh, on anything of that same, I guess, solution. So BSA and proteins to get, if you know the absorbance, you can get the concentration of the unknown. Okay, so I will post that also. And if you have any questions, please email me. Other than that, please read the book, please read the experiments, and give me a nice A plus paper. All right, guys. See you later.